Here we are with term 2, very small things for year 7. First we looked at the atom. It's got 3 subatomic particles. It's got your electron, your proton and your neutron. Electrons buzz around the outside. Your protons or your neutrons are found in the nucleus. So the positive is a proton, the neutral is a neutron, and then your electrons are negative. Relative mass, all the mass is in the nucleus. So your protons and neutrons, and then you've got your electrons, which are tiny around the outside. Lesson 2 then was on elements, mixtures and compounds. We need to know that an element is made up of one type of atom, or sometimes we say of one type of molecule. So they're the same colour. Whereas a compound needs to be made up of 2 or more elements. So you can see that 2 different colours stuck together. They're chemically bonded, they're hard to separate. Whereas a mixture is a mixture of everything and it's easy to separate. So we could have some element, we could have some molecules, we could have some compounds, but not joined together. Easy to separate. Lesson 3 was on electron structure. So we can see that electrons always occupy shells, and there's rules to these shells. We always fill the lowest level first, so the first circle, and the first circle can hold 2 electrons. First shell, 2 electrons. Then we create another circle, and that can hold up to 8 electrons. And then the third shell, another 8 electrons again. They're always happy in full shells. Lesson 4 was the microscope, and we need to be able to label this. So let's start with the eyepieces where we look through. We've got the body tube. We've got the stage where our sample goes. We've got the mirror which reflects light up. Sometimes this is a light. We've got the handle to pick it up. And then we've got these 2 little knobs. We've got the fine focus and the rough focus. The rough focus is the knob that you will turn to get some sort of image, and then you'll sharpen the image by using the fine focus. There's often different lenses as well. So we've got a high and low power lens here. These are called the objective lenses. You also need to know how to prepare a slide. And we'll do that using a cover slip. Lesson 5 was on cells. 2 different types of cells we're going to learn about. One is an animal cell and the other one is a plant cell. Now a plant cell has everything an animal cell has, plus another 3 things. Let's look at these. So things that's familiar with both is a nucleus, cytoplasm, cell membrane and mitochondria. We need to know that the nucleus holds the DNA. It controls what the cell does. Cytoplasm, where chemical reactions happen. The cell membrane allows things to pass in and out. And the mitochondria is the site of respiration. It's where energy is created. Now a plant cell has all these things, plus 3 more. It's also got a cell wall to give it support and structure, a vacuole, which is filled with cell sap, and chloroplast, which is the site of photosynthesis. Photosynthesis in another lesson. Lesson 6, we looked at DNA. And for this, we only really need to know that adenine, A, pairs with thymine, T, and guanine, G, pairs with cytosine. And these will always be paired together. And we call them base pairs. This backbone is a sugar phosphate backbone. And that was that lesson on DNA. Next one was on waves. We need to be able to label a wave diagram. So, let's first count how many waves we got. We've got 1 full wave starting here, goes all the way up, down halfway, all the way down, up, and that is 1 full wave. So we can see that there are 1, 2 waves here. We call the distance from there to there a wavelength. But the distance from there to there could also be a wavelength because it's one full cycle of a wave. The distance, the maximum displacement, is called the amplitude. The top of the wave called a crest, or we can sometimes call it a peak. And the bottom of the wave is called the trough. And that was waves. And then we're going to use the triangle to calculate wavelength again. So we've got V, which represents wave speed, F, frequency, and lambda, this symbol, represents wavelength, which is measured in meters. Frequency was hertz. And then because this is a speed, it's meters per second. And again, I would use my finger to cover the letter that I want to find. So if I want to find frequency, I would cover my finger, and it's V divided by lambda. And that was term 2, very small things.